So now we get to figure out species and speciation. If this video sounds a little odd, it's because I had to re-record it literally at my house instead of in front of students. I may have forgotten to push the record button in class, so you won't get any cool comments on the side from any students, and I probably won't go into terribly many, off-track stories. So maybe this one will go a little faster. Today we're talking about speciation, continuing our discussion of pre- and post-zygotic sexual isolation. So first up, we're going to talk about pre-zygotic sexual isolation. This happens before the zygote is formed. So anything that interrupts the mating process, anything that stops the sperm from fertilizing the egg, that's termed pre-zygotic sexual isolation. And on the screen, we're going to go through a lot of examples, but on the screen you can see the sea slug and the common field cricket. Now the sea slug's super interesting here because sea slugs will emit a pheromone. And that pheromone sort of tells the other sea slugs around it, hey, not only am I willing to mate, but it gets them in the mood to mate too. Now, if one sea slug in this population had a mutation that did not allow it to receive that signal, or it changed the signal in some way that it emitted so the other sea slugs didn't recognize it, it would stop the ability for that slug to mate. That would be a pre-zygotic isolating mechanism if there was a change in that pheromone. For crickets, have two things they need to worry about. They've got to be ready to mate at the right time, so whether in spring or in fall or in summer, and they need to have the right song. If the song is wrong or the cricket sings its song at the wrong time, it's not going to be able to find a mate. Those are pre-zygotic isolating mechanisms. Pre-zygotic measures happen prior to the fertilization of the egg. So anything that stops fertilization of the egg. Here you can see a bird doing a little dance. Birds do these great mating displays, and they are very, very specific to the species. You probably don't find this mating display terribly attractive. You might find it hilarious, but not terribly attractive. The birds of the same species, though, will find this attractive and will hopefully allow this bird to mate. If the bird does the dance wrong, gets the behavior incorrect, that becomes a pre-zygotic isolating mechanism. So we've got 5 big pre-zygotic isolating mechanisms. There's habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. And behold a list. I hate throwing lists at you and then saying, hey, just remember these things. So I'm going to give you several examples of each of these distinct mechanisms to hopefully help you remember them. But really, they just make sense. For example, habitat isolation. If there's a geographic barrier that stops you from breeding, you've got habitat isolation. You who live, I don't know, in the United States may find it very difficult to mate with somebody who lives in France. There is an ocean separating you. That's an isolating mechanism. You've probably experienced this if you have like a long distance relationship. There is definitely something stopping you from mating. There are ways to overcome it. There could be some migration, but in general, habitat isolation will keep two populations apart. Sort of a generic example is shown here. You can see there's this. There are two populations, this blue and this sort of, I guess, a greenish blue or bluish green and then a greenish blue on the bottom. If those two populations get separated by a river and that river stops these two populations from coming into contact, that leads to isolation, an eventual speciation as those two populations end up breeding within themselves and the alleles in those distinct populations end up going to fixation. So you have two separate populations eventually going on to speciate. Here's an example of habitat isolation. Fruit flies. Fruit flies love rotting fruit. I don't know if you've seen them flying around your kitchen or whatever, but they love rotting fruit. On screen, you can see two types of rotting fruit. You've got a mango and you got a banana. Some fruit flies prefer to mate on the mango. Some fruit flies prefer to mate on bananas. And the two don't come together. It's almost like, I don't know, meeting friends. Some of your friends like to hang out at one place. Some of your friends like to hang out at some place totally different. I don't know. Some of them might like to hang out at clubs or bars or something, and others like to hang out in libraries. And you might not get much overlap. I'm not trying to pigeonhole you, but typically you're going to see two distinct populations. In this case, the fruit flies that prefer mangoes mate with fruit flies who prefer mangoes because fruit flies, unlike you, perhaps, prefer to mate on their food. So the mango fruit flies mate on the mango, the banana fruit flies mate on the bananas, and it ends up creating two entirely separate isolating mechanisms. Back in the 1950s, they decided to explore this idea. So what they took was one population of fruit flies, one interbreeding species of fruit flies, and they separated them randomly into two groups. Into one group, they put it into a box and had it live on purely maltose, maltose-based foods. Into the other box, they made it based. They created starch-based foods. Now, again, these fruit flies live and mate on their food. They love the food's center of their life, like it is with you. It's the center of their life. So after many, many, many generations, those flies that lived on the maltose-based food may have developed some unique biochemical markers and maybe some unique behaviors, and they tended to prefer to mate only with flies that lived on the maltose, while those who were on the starch-based foods preferred to mate and reproduce with those that are starch-based. When you brought the two populations back together, you saw that continue. So even when you had an original population that could mate together, over time, you separated them, you gave them different environments, different food in this case, and you bring them back together, you find that they tended to mate back with the group that they were assigned, not back to randomly within the population. And it showed how, this was a classic example that showed how habitat isolation can eventually lead to reproductive isolation and speciation. Another type of isolation is temporal isolation. Temporal isolation is where you are not ready to mate at the same time as the other members of that group. This could be due to the timing of mating cycles. Interbreeding between two populations might not occur. So even though you may have the same species, if they're ready to mate at different times, you get pre-zygotic isolation. They could mate if they were together at the same place, at the same time, and ready to mate, but they're not. It's similar to, I don't know, if you were ready to mate, but your partner wasn't. That's not. There's a disconnect, and you're not going to end up mating. Or I don't know. Abraham Lincoln died a long time ago, and you're still alive right now. You're not going to be able to mate with Abraham Lincoln because you're isolated by time. I don't know why I chose Abraham Lincoln, but there you go. Enjoy that image. Here's an example of temporal isolation. It's a frog. If you go outside in the spring or in the fall, you can hear frog song. These frogs are chirping their little hearts out in order to make sure that they can mate. They're displaying, if they've got the right sound, then other frogs will be attracted to that. Now, some of these frog sounds you'll notice are very, very deep. Some of them are very, very high. But the important thing to note is that in spring and in fall, you have entirely different sounds. That's because the spring frogs are trying to mate with other spring frogs, and the fall frogs are trying to mate with other fall frogs. It may be possible for them to mate, but the spring ones only prefer to mate in spring, and the fall ones only prefer to mate in fall. They are temporally isolated. The most classic example, and the one that probably impacts us the most in Virginia, are the cicadas. Yes, that loud insect that goes up into the trees and just screams for what seems like months on end during the summer. are a great example of temporal isolation. These cicadas hatch in broods. They live most of their lives underground. As they're underground, they mature. They're just sort of larvae that over time grow, get bigger. You may have pulled some up when you got a shovel full of soil sometime. But then, once every several years, specific to the population, they will emerge from underground and begin to they'll shed their exoskeleton, grow some wings, and be prepared to mate. Now, there are cicadas that come out every year. There are cicadas that come out every 3 years, every 5 years, every 7, 11, and I think 17 years. The 3-year cicadas are only going to be able to mate with the 5-year cicadas every 15 years, lowest common denominator. So these two populations are in general temporally isolated from each other. They are, even though they're capable of mating with each other, they are coming out at different times and ready to mate at entirely different times. Crickets, again, you've got summer crickets and you've got fall crickets. Summer crickets will chirp one way at a specific time. Fall crickets will chirp at a completely different time. It's only through that signaling at a specific time will these crickets be able to mate. The fall crickets are temporally isolated from these summer crickets. And we talk about chirping and chirping and stuff with temporal, but chirping is really a behavioral isolating mechanism. Different populations have different ways of signaling mate choice. So you've got to let the other organism know that you're ready to mate, and they then also need to recognize that signal as a preparedness to mate signal. For example, birdsong. Birds sing for exactly two reasons. You go out in the morning, you hear birds singing, you're like, oh, it's beautiful, the birds are singing. The birds are either singing, I hate you, this is my area, leave. It's a territorial sign. Or they're singing, hey, check me out, I want to mate. Those are the reasons birds sing. This is my place, or I want to mate with you. As you can imagine, if you get the song wrong, or as birds get the song wrong, it could signal entirely different things. Let's say that a bird is singing trying to mate, but the other bird doesn't recognize it, so they see it as a territorial sign. It means, one, the bird's not going to be able to mate, and the other bird is going to try and fly away. Or just the opposite, if you've got a bird who is trying to defend their territory and another bird recognizes it as a mating song, I guess there would be some sort of comedic misunderstanding. Insect mating dance. This is another very well-studied behavioral isolating mechanism. In fruit flies, I used to study fruit flies. So, surprise, I studied fruit flies. And specifically, when I was doing my first master's degree, I worked on looking at the mating habits of Drosophila ananassi. And so, yeah, that was super fun. You just sit in the lab and literally watch fruit flies mate. It was awesome. But they all went through the same process. They would orient themselves. The male would sort of come up right onto the side of the female and then go in front of her and then off to the other side, and then in front of her and off to the other side, sort of say, hey, check me out with all of your eyes. And then he begins to sing. Now, we can't really hear his song. He flaps his wings really hard, and that vibration is at a certain frequency that makes the female receptive to mating. Now, if he flaps his wings wrong, it means that, no, there's not going to be any mating. He didn't hit the right frequency, and she just ups and flies away. After she seems receptive, the male begins to follow her, which kind of seems a little stalky, but apparently it works for flies. He follows her around for about 15 to 20 seconds. The male then begins to lick the female's genitalia. That's important for these flies. If he licks her wrong, there's not going to be any mating. And then there's actual copulation. The copulation mechanism, he can't go too fast, or else the process will the female will reject him. If he goes too long, the female will also reject him. If those behaviors don't line up to the female preference, mating doesn't happen. And then finally, it's over. They go their separate ways. If the male does not follow through these behaviors step by step every single time exactly perfectly, they will not successfully mate. That stops the zygote from forming. This is pre-zygotic. For humans, eye contact is could be a behavioral isolating mechanism. In different cultures, eye contact means very, very different things. For some cultures, eye contact is a challenge. For others, eye contact is a way of showing that you're engaging with your listener. And in other cultures or at other times, eye contact could be flirtatious. It could be a signal of mating. I don't know. I don't know what's going on between these two on the screen, but eye contact could be many, many different things, and it's up to the culture and interpretation of the observer to recognize this as a mating behavior or a territorial behavior or just a listening behavior. Mechanical isolation is a little bit easier to understand. Basically, with mechanical isolation, there are morphological features of the organism, so what they look like, that stop them from interbreeding. For instance, look at these two dogs. It'd be difficult for the smaller male dog to mate with the larger female dog. I guess he'd need like a step stool or something. There's mechanical isolation there. The parts just don't line up without assistance. These two copepods. Copepods are little, sort of think of them like water bugs. They've got jointed segments on their bodies, and their genitalia are hidden behind some of those segments. If they don't have segments that match up exactly where the genitalia are, there's not going to be a mating won't be able to occur. There's literally morphological parts that don't line up. This would stop the zygote from forming. And then the last category is gametic isolation. So if we got through all of the other isolating mechanisms, you're in the right place at the right time, you do the right behaviors, all the parts line up, all the stars have lined up, and you successfully mate. The sperm is on the way to the egg. With gametic isolation, the sperm and the egg simply don't fuse. They don't come together. One example are sea urchins. Sea urchins will, in general, just sort of put up a cloud of gametes. And in this cloud of gametes, the sperm and the egg will come together. They do this all at once. It's kind of neat. It's based on, I think it's based on salinity in the ocean water at a certain time of the month, at a certain time of the year. And suddenly, there's this cloud of gametes in the water above the sea urchin colonies. Eggs and sperm go to find each other. Sometimes, if you've got different species of sea urchins, those gametes run into each other, but they don't fuse. There's some chemical change, some chemical barrier that stops that. So with gametic isolation, you get the sperm getting to the egg, but not being able to fertilize the egg. Thus, pre-zygotic, before the zygote forms. So now we got all those pre-zygotic isolating mechanisms. We look at post-zygotic isolating mechanisms. So let's say the sperm fertilized the egg and you've got a zygote forming. That's not the end of the story. You could still have post-zygotic isolating mechanisms. After the zygote forms, there may still be issues that prevent future generations. With post-zygotic isolation, usually the issues come about due to the X chromosome. This guy named J.B.S. Haldane, he's the gentleman on the left there, he said that in the F1 offspring, so the first generation of two different animal races, so two organisms coming together that are different, and one sex is absent, rare, or sterile, that sex is usually the heterozygous or the heterogametic sex. In humans, males are the heterogametic sex. We have an X and a Y chromosome, so two different sex chromosomes. In butterflies, it's just the opposite. Females are the heterogametic sex. Basically, what that says is there's a large X effect. The X chromosomes has an outsized influence on the viability of sort of these mixed progeny. The reasoning behind that is that X chromosome is very, very large. It's got a lot of genes on it. The Y chromosome is small, it's got some genes, but really it's just a lot of leftover genetic crap. So if there's a problem in the X chromosome, the Y chromosome cannot compensate for it. Whereas with other organisms, with the homogametic sex, the other chromosome could compensate. So if there's a problem, it's usually going to be due to that X chromosome issue in hybrid offspring. This could lead to hybrid inviability. That means that the zygote formed, it begins to develop, but it does not ever fully develop. It does not survive to then itself try to reproduce. It dies. So you've got a zygote form, but it can't continue. There's some developmental errors, and it cannot continue, and it doesn't produce a living organism. Sometimes you have these hybrids forming, and they do produce fully functional living organisms, but those functional living organisms are themselves sterile. This is called hybrid sterility. So hybrid inviability is where the organism cannot survive to reproduce. Hybrid sterility is where the hybrid could try to reproduce, but it cannot produce an offspring itself. An example of this is a horse mating with a donkey, so two different species. They will produce a mule. That mule itself lives. It's a strong worker. But the mule is sterile. It cannot produce offspring on its own. So that sort of reinforces these species boundaries between horses and donkeys. Now, pre- and post-zygotic isolation are connected. Pre-zygotic isolation will usually occur before post-zygotic isolation. And that's not because pre means before and post means after. It's because pre-zygotic isolating mechanisms are typically behaviors or conditions that will occur in a short time frame. So with a pre-zygotic isolation measure, behaviors change relatively quickly. Look at the way we behave now compared to the way we behaved 20 years ago, compared to the way we behaved 50 years ago. Heck, just with humans, the pickup lines from the 1950s or the attitudes of the 1950s probably would not help you get a mate in today's day and age. In a short period of time, that behavior, the equivalent of a bird mating dance, has changed so much that it would stop reproduction. That being said, our genetics that would lead to post-zygotic isolation have not changed terribly much. Genetics changes very slowly compared to behaviors. So those post-zygotic isolating mechanisms are going to be very, very slow to change compared to pre-zygotic isolating mechanisms. Now, if we have two different populations, and they've been separated for a long period of time, either pre-zygotic or post-zygotic isolation will arise in that population. Think of Darwin's finches. When we see this happen, it is usually, far more often than not, pre-zygotic isolation that occurs first. It's a change in a signal. It's a change in a behavior. It's a change in the timing that will occur prior to changes in the molecular mechanisms associated with the zygote's development. So pre-zygotic isolation usually occurs before post-zygotic isolation. And then the pre-zygotic barriers, those those behaviors and that timing is going to then reinforce species boundaries. So it may be a very critical step to giving the populations time to diverge genetically to become two true, totally separate species. If we relate this back to the biological species concept we already talked about, a reduction in fitness among hybrids can be an initial cause of speciation. Basically, if a hybrid has sterility or inviability, that serves to stop mixing of genes and keeps two populations apart. So there we are. We've gotten through pre-zygotic and post-zygotic isolation. Sexual isolation could be pre-zygotic before the zygotes formed in the form of temporal isolation, geographic isolation, gametic isolation, behavioral isolation, and so on. Anything that prevents the egg from being fertilized, that's pre-zygotic. Post-zygotic isolating mechanisms like hybrid sterility and inviability occur after the egg has been fertilized, and typically you will get pre-zygotic isolation before post-zygotic isolation, but both of them will reinforce species boundaries.